Okay, so is my screen visible?
Yes, sir.
Good. So it all began, that means understanding how the information is processed inside the organism. It actually began much earlier than that. Before understanding this aspect, that okay, there is some. Just give me a moment. Okay. Yeah. So before it was understood that the information is processed inside the organism, you should also know that there is something which is storing the information and passing that information from one generation to other, right? So, in other words, it was something like to predict that or to prove that the information bearer is DNA, or the biomolecule which carries the information from one generation to another is DNA. So there's a landmark experiment which actually laid the foundation of understanding that DNA is the genetic material. So can anybody tell me what things were actually done during this experiment? Because you might have learned about this Griffith's experiment in your first year of microbiology, and perhaps you might remember that what all was done by Griffith's group in order to understand, or in order to get a path ready for predicting that DNA is the genetic material. So can anybody tell me that how this experiment was conducted? It is actually in front of you, everything, but still, yes. Does anybody remember this experiment, Griffith's experiment? This is a landmark experiment that was done. Yes. Anyone?
I can try, sir.
Yeah. Okay. Go ahead.
So, Griffith saw two types of colonies of Streptococcus pneumoniae, and
Yes.
the difference he saw with his physically, the difference he saw between the cells were one colony of cells were had a glassy, shiny finish over the cells, were a little bit shiny, and the other one was not that much shiny. So one colony he called as smooth colony of cells, and the other one he called as rough strain or R strain of cells.
Right.
And then so first he uh, injected uh, the smooth colony of cells into the mice, and he saw that the mice died, and then he injected a rough strain of cells, and he saw that the mice lived. So then he started thinking about it, that how is that possible. So he tried another experiment to check that how this is happening, why there is different effect after ingesting different after uh, ingesting different types of bacteria. So he did the smooth strain of cells, the S strain of cells, and then he injected those cells into the mice, and then he saw that the mice still continues to live. So he thought that there might be some difference between the cell wall of the cell, or the cell uh, plasma membrane. So
Okay, if you allow me to interject here, uh, you missed one part of the experiment that I was trying to highlight. And the thing was like, uh, you were good in the first part, that there were two different forms of colonies that were observed for the same organism is Streptococcus pneumoniae, which is responsible for causing pneumonia. Okay, so there were two forms of colonies observed. One was having a smooth appearance, smooth in the sense glossy kind of an appearance, and the other one was having a rough appearance. The colony, it's not the organism. Okay? So when you plate the organism onto an agar plate, you can see the morphology of the colony as well. Right? It's one of the most preliminary observation about the organism's nature that you're looking at. So he observed that there were two different types of colonies for the same organism. Okay, one was smooth, one was rough. The smooth colony, when organisms were taken from the smooth colony and were used to infect the mice, then the mice developed pneumonia and the mice died. However, when the organisms were taken from the rough colony and were injected into mice, then the mice lived. Now, there was something else that he tried. He tried to heat kill the S strain. S is the infective strain. He heat killed it. That means he actually used the culture and he boiled the culture. Okay, that essentially means that the organism is done. That means it's not there. Now, what next? How we can take it?
so after this, uh, I was saying that he thought of that there might be something in its outer covering of the cell, that is the plasma membrane of the cell. So he tried putting up live, uh, that means not healed killed, just live R strain and heat killed S strain together into the mice, and then he saw that mice still dies. So what we uh, what he uh, uh, predicted from this was that uh, even after uh, so first of all, even after putting heat killed S strain, the mice continues to die. Now how is this possible? Because when heat killed S strain was put, then mice continued to live. So the so he predicted that when the R strain was inside the mouse and it was growing inside the mice, it was making new generations, it picked up something from the heat killed strain, and that something he infested into itself, and he continued to reproduce, continued its growth, bacterial growth. Hence it became virulent and caused pneumonia and killed the mice. And
Right.
this something
So,
he
in
called.
one way, can I put it like this? Okay, for everyone to understand and to keep pace, can I say this, that there was some molecular factor that was involved in this process, so that which was retained even after the organism was heat killed. And then when you mix that heat killed concoction with the live strain, which is non-virulent R strain of the organism, then the R strain of the organism was capable of transforming itself into a virulent strain. Is it okay if I say this?
Yes, sir.
Okay. So now, why I'm saying this, why I'm putting it in a slightly different manner compared to what you said, Pavika, is because there was an interesting stuff happening. Once the organism died, that means once the mice died, then he isolated from the dead mouse both the strains. Quite an interesting stuff, right? That means why we are saying that the organism got transformed. That means the avirulent form of Streptococcus pneumonia got transformed from avirulent state into a virulent form is proven here that although you have used a concoction of heat-killed S strain, there was something there in the S strain that was capable of converting the R strain into the S strain. Is it good if I say this?
Yes, sir.
Okay. So now if we say this, then what is your inference about the entire experiment? What you can infer out of it? See, now why I'm asking it is because we already know that now the DNA is the genetic material, right? At that point, it was not known. It was in 1928 that the experiment was performed. There was no knowledge about DNA being the genetic information bearer, right? So now, if there is no way that you can decipher that, okay, there is one molecule which is capable of informing the R strain how to become virulent, then how do you infer this experiment? That means I am completely unaware of the fact that DNA is the information bearer. There is no way that I can actually predict that there is anything within the organism that can carry the information from one generation to the other, or rather can make the organism self-sufficient and convert it from an avirulent form into a virulent form. Is there any background now? There is no such background. So what kind of an information or what kind of an inference you can draw out of this experiment? So one by one, we will take some points on the basis of whatever you have understood about this experiment. Is there anybody still remaining in the group which doesn't understand how the experiment was conducted? Okay, because there is silence, I believe that you have understood. See, one thing again I'm repeating. If you have any doubt in understanding this experiment, or any experiment for that matter that we are going to discuss all throughout this session, please let me know it right in the beginning, because if it is not told to me that you are not understanding it properly, then you will not find it interesting at all. Okay. So now, in case there are any doubts, we can refer to the doubts, or else you start inferring out from this experiment. What do you infer? The major takeaway from this experiment. Okay, let me try to put it and you will tell me whether this you agree with me or not. Okay. What if I say this, that from the Griffiths experiment, one thing is for sure there, that even when the organism is dead, when you heat kill it, even when the organism is dead, something remaining, so perhaps it's a molecular factor. We will not refer it as any molecule for at this moment. So there is a molecular factor it still retains this capacity of converting the rough form into the smooth form. That means avirulent form into the virulent form. Do you agree?
Yes, sir.
Okay. And one more important aspect of it is that when you are heat killing the organism, this molecular factor doesn't lose its ability, right? It's a somewhat stable molecule. Is it good if I say this also? Yes?
Sir, can you please repeat this statement?
Okay. So one thing is certain that you have a molecular factor which is capable of transforming the avirulent form into the virulent form. Another what you can say inference that I can draw from this experiment is that this molecular factor can to certain extent resist heat, right? So can I say that the molecular factor is stable enough?
Yes, sir.
Okay, good. So now there are two things about the molecular factor that we know on the basis of this experiment. One is it is capable of transforming the avirulent form into the virulent form, and two, it can resist heat, or we can say that it's stable. Good to go ahead.
Yes, sir.
Okay, fine. Now you haven't understood that what is this molecular factor as of now, right? So there were, on the basis of the experiments and observations that were recorded by Griffith's group, it doesn't in any way establish any background related to DNA being the information bearer. So it's still not there. Okay, it's just a molecular factor. So we are not very certain that what is this molecular factor. So obviously the next set of experiments should be directed to understand that what is this molecular factor, right? Yes or no?
Yes, sir.
Okay, good. So can you tell me what was the next experiment that was planned in order to answer this question?
Sir, Avery, McLeod, and McCarthy experiment.
Yes, it was. Okay, Pooja, can you let everybody know that what was this experiment?
Sir, I can try.
Okay, no problem. Go ahead.
Sir, sir, they mixed S strain of DNA with
Okay,
मतलब जो solution था उनका उसमें they added
right.
DNA first. Sir, DNAs know that DNAs degrade or destroys DNA. So there will be no transfer of that virulent gene.
Okay,
Okay. And when they added RNAs to the solution, it destroys RNA, but still the transformation was there. That into R strain into S strain.
okay,
And when they added protease in that solution, protein degraded, but DNA अभी भी intact ही रहा, तो DNA there will be transformation still. So
okay,
from this experiment, they concluded that DNA was the transforming principle and not protein or RNA.
okay. There is a slight bias that you are adding to the story. And that bias is because you already know that DNA is the information bearer, right?
Yes,
Can I
sir.
say this, Pooja? Because
Yes.
you already know that DNA is the genetic material and it's the information bearer. Imagine that you are at the place of Avery, McLeod, and McCarthy. There is no base that can tell you that there is DNA which is the information bearer. Okay. You only know that there could be 4 different biomolecules existing. So instead of saying that it is DNA, let me say it is nucleic acid. It includes both the DNA as well as RNA. Right? There is protein, which is another important biomolecule. There is carbohydrate. There are lipids.
Super.
Right? So now you need to address all of them together, correct? Because you don't know which is the information bearing chemical entity, right? So the experiment that was designed was to understand which of these 4 is the information bearer. What do you think that went wrong in the experiment? First of all, let me first address this question, that is everybody okay with the experiment? That means they know what they did in the Avery's McLeod McCarthy's experiment. Everybody knows that what was done there.
Yes, sir.
Okay. So first step first. Yes. See, because you are taking in the previous Griffiths experiment, because you are taking the concoction of the organism which is heat killed, right? Okay. Now the organism is heat killed, and you know that there could be a stable molecular entity which is still there and is capable of bringing about the transformation. Fine. So now with all these 4 with you, how will you proceed? It's written over there. I don't need to actually tell you how it was done. And because you already know the process. So first step given over here.
No.
Yes.
So they first removed lipids and sugar from the solution of heat killed S cells
Okay. How did they
because
remove
lipids and sugar are not stable molecules.
lipids and sugars are not stable molecules? That's what Tanaya is saying. How many of you agree? Yes. Yes. Nobody agrees with you, Tanaya.
Sir, not sure about it.
Not sure about it. Okay. Fine. To a certain extent, what Tanaya said is true. That instead of focusing their understanding about on carbohydrates and lipids being responsible for transferring the information, they actually tried to focus more upon proteins and nucleic acids. Okay. So what could be the reason behind it? Why is it so that you need to focus only on proteins and nucleic acids? Tanaya answered it partly because carbohydrates and lipids are not that stable compared to proteins and nucleic acids. Okay. Rather, if I put it other way around, when the molecular entities that were observed to be present in the concoction of the heat-killed organism, it was essentially containing proteins and nucleic acids. It was not containing carbohydrates and lipids to a great extent. So that rules out the presence or the activity of these two biomolecules being associated with information carrying capacity. If there are no carbohydrates and almost negligible lipids, what essentially remains in the concoction are these two molecules, nucleic acids and proteins. So they first tried to address the issue proteins, understanding that whether they are capable of bringing about the transformation. So what they did in order to find out whether proteins are the ones, they remove the protein from the extract by organic solvent method, and that they found out that the extract still transforms. Okay. The first step is quite interesting in the sense that in the extract you have only two molecules, and out of that you are getting rid of proteins, meaning you are enriching the stuff for nucleic acids, right? So one thing becomes certain, that perhaps nucleic acid retain the capacity, right? Also, they found out that it is not that the nucleic acids alone can perhaps do it. It's a different way of looking at it. Okay. They digested the proteins in order to be completely sure that there is no protein remaining in the concoction. They used the protein digesting enzymes, which will completely destroy the protein. And that too didn't cause any change to the transforming principle. So perhaps now it is more or less certain that it is nucleic acids. Also, they found out that when you add an RNA degrading enzyme within these two now, what remains is DNA or RNA. Right? So when they added the RNA degrading enzyme, then they found out that it still retains the ability to transform. So basically, they are going in what is called as a bottom to up approach, in the sense that they are removing one molecular entity at a time, or using some way by the way of which one molecular entity can be removed, and then whatever that remains in the concoction, whether that is capable of transforming. Do you think that is a good approach to go for it?
Yes, sir.
Yes, sir.
Okay. Finally, they used DNAs, and which breaks down DNA as well. And then they found out that the transforming ability is lost. Is it not a good enough proof to say that DNA is the information bearer?
It is, sir.
It is. Then why is it so that we give all the accolades, or all the accolades were actually given to the Hershey and Chase experiment? All the credits are given to Hershey and Chase experiments, an experiment that were conducted, which actually completely confirmed that the molecular entity which is capable of transforming the information, or bringing about transformation and carrying the information from one generation to other, is DNA. Why is it so? Is there a need of the Hershey and Chase experiment? What do you think? That there is any need now after every experiment? Yes, Avika?
Yes, sir. Because we know that DNA is the transforming principle, but
Right.
we don't know how it is getting transformed from a dead cell into a live cell and making more live cells of the S strain, which were already dead.
Mhm. Is that the way that you look at it? Because here there is no way by the way of which the genetic material, whether it is DNA, whether it is protein, whether it is RNA, there is no way by the way of which you can predict that how the transformation is happening. And that is not the, what you can say, aim of the experiment. The aim of the experiment is just to prove what is the transforming principle. How is it happening? That's a different story altogether. What say?
But sir, you asked that why did we need Hershey and Chase experiment?
Yes, because the question is simple. See, if every MacLeod and McCarthy's experiment is good enough to prove that DNA is the genetic material and it is the information bearer responsible for transforming the non-virulent strain and converting it into virulent form, why there is a need for Hershey and Chase experiment? And let me tell you, Hershey and Chase experiment is a greatly lauded experiment. That means it is given more credits compared to Avery, MacLeod, and McCarthy. Although the foundation is laid by Avery, MacLeod, and McCarthy's experiment, but some way short, it's not complete. Why do you think so? That's what my question is. Why there is a need of Hershey and Chase experiment to confirm that it is DNA? Okay, let me ask you this question. Say, for example, if you are into some kind of a scientific endeavor and you are trying to explore something, will you try to explore the same thing which was done by some of the people previously? Will you try to replicate it just, or will you try to address the question again, just in a slightly different manner? Is it needed?
Yes, sir.
Why so? Why is it needed, Pooja?
Sir, just from one experiment, we cannot be very sure that DNA is the transforming principle. Maybe this could be reason.
Okay, so if you want to challenge the previous group, then you should have solid foundations for that, right? You
Yes.
should be able to say that you did this wrong. And that is why I am planning another set of studies which will confirm that, okay, what you said is correct. Is it good enough to say this?
Yes, sir, maybe.
So what do you think was done wrong by Avery? Yes, anyone? Do you think that there is any flaw in the experimental design that was used by Avery, MacLeod, and McCarthy to prove that DNA is the information bearer? Yes, we are nearing the end of the class. So what if I do this? What if I give you the entire day to think over it? Okay, and we'll address this question again tomorrow, and then we'll go into Hershey and Chase experiment. Is it okay?
Yes, sir.
Yes.
Okay, good. So you can just ponder over the same experiment of Avery's, MacLeod, and McCarthy, and find out, are there any flaws in the experiment? And why is it so that there is a need of Hershey and Chase experiment to conclusively say that, okay, DNA is the information bearer and it's the genetic material. Fair enough? Now I will stop sharing. And in case you are having any doubts in today's session, then we can discuss it. And one last thing, why I have why I'm closing it early is just wanted to know from your side that are you finding this way of learning uh, okay or not? Is it going in sync with your way of understanding, or do you want me to change it a bit? Because later on, the situation will be like we will be addressing those experiments which you haven't heard about previously. These are the experiments that you have heard about, right? Later on, we'll talk about those experiments which you haven't heard about in order to find out that what exactly happens during transcription. So do you find this way of learning, whether it is suiting you or not?